So, transport of carbon dioxide in a healthy human adult under resting conditions produces nearly 330 liters of CO2 in 24 hours. The CO2 formed in tissues diffuses to blood. Blood transported into lungs where it diffuses out from blood and escapes to outside during expiration. Blood transports CO2 from tissues to lungs in three ways: carbonic acid as carbaminohemoglobin and bicarbonate. So, transport of CO2 as carbonic acid. In this case, CO2 reacts with plasma water and forms a weak carbonic acid. This occurs in tissue sites. On reaching the lungs, carbonic acid dissociates and releases CO2. In man, about 5% of the CO2 is transported in this manner. The remaining 95% is transported by RBCs. So CO2 plus H2O giving H2CO3 plus H2O plus CO2. Okay. The formation of H2CO3 in blood plasma is influenced by the solubility coefficient and tension of CO2, PCO2, and the internal temperature of the body. In many invertebrate species, this reaction is catalyzed by the enzyme carbonic anhydrase present in their blood plasma. In vertebrates, this enzyme is present in the RBC. So, the formation of H2CO3 in plasma is a very slow process in carbohydrates. So, when we learn about this, transport of CO2 as carbaminohemoglobin is very important. In this case, CO2 first diffuses from plasma to RBC, then it reversibly combines with the amino NH2 group of the globin part of hemoglobin and forms carbaminohemoglobin. The formation of carbaminohemoglobin is greatly influenced by the high PCO2 in the tissue capillaries. So, also its extent depends on the availability of NH2 groups and pH and PCO2 of the blood. On reaching the pulmonary capillaries, where PCO2 is very low, carbaminohemoglobin dissociates, releasing CO2. This dissociation is catalyzed by carbonic anhydrase. The CO2 soon diffuses to pulmonary alveoli and then escapes to the outside. In man, about 10% of the CO2 is transported in this manner. CO2 plus HbNH2 gives HbNH gives HbNH2 plus CO2. Then transport of CO2 as bicarbonates, isohydric transport. In man, about 85% of CO2 is transported in chemical combination as bicarbonates. This occurs with minimum change in pH and is therefore termed isohydric transport. In this case, most of the CO2 at first diffuses from blood plasma to red blood cells due to the high PCO2 in the plasma. In the RBCs, it reacts with water and forms carbonic acid. In vertebrates, this reaction takes place more rapidly than in plasma, since it is catalyzed by the enzyme carbonic anhydrase, which is absent in the blood plasma of vertebrates. Carbonic acid soon ionizes to hydrogen ions and bicarbonate ions, HCO3 minus. Then CO2 plus H2O, carbonic anhydrase, H2CO3. H2CO3 again dissociated to H plus plus HCO3 minus. During the ionization of H2CO3, the intracellular pH of RBCs falls low. The oxygen affinity of Hb decreases and CO2 gets unloaded from Hb. Thus, the release of O2 and the uptake of CO2 by Hb occurs simultaneously. In the blood, the ratio of CO2 to H2CO3 is approximately 1000 raised to 1, and that of CO2 to HCO3 minus is about 1 to 20. Bicarbonate is perhaps the predominant form of CO2 in blood at normal blood pH. The hydrogen ions formed by the ionization of H2CO3 are soon buffered by deoxyhemoglobin Hb, which acts as a proton acceptor. They immediately combine with deoxyhemoglobin, oxygen-free hemoglobin, and form hemoglobinic acid. The release of H plus ions and their immediate buffering by deoxyhemoglobin is called Haldane effect. After Haldane, the release of H plus ions and their immediate buffering by deoxyhemoglobin is called Haldane effect. At the same time, bicarbonate ions diffuse to the plasma from red blood cells when their concentration increases within the RBCs. This outward diffusion of HCO3 minus from RBC to plasma momentarily upsets the ionic equilibrium and the electrical balance between plasma and red cells. To regain this lost balance and also to establish electrochemical neutrality, an equal number of chloride ions Cl minus diffuse into the red cells from plasma. This is known as chloride shift or Hamburger effect or Hamburger phenomenon. What is it? To begin regain chloride effect. Okay. So free chloride ions are made available by the dissociation of NaCl or HKCl in the plasma. Inside the RBC, chloride ion combine with K plus and form KCl. The HCO3 minus ions in the plasma combine with Na and form NaHCO3. H plus plus Hb giving HHb inside RBC, NaCl, Na plus Cl minus in plasma. Cl minus plus K plus KCl inside RBC and HCO3 minus plus Na plus NaHCO3 in plasma. Summary of the transport of CO2 as bicarbonate in the blood of tissue site. CO2 diffuses into RBC from plasma. CO2 plus H2O giving H2CO3 in RBC. Then H2CO3 gives H plus plus HCO3 minus in RBC. Then H plus plus HHB in RBC. That is the Haldane effect. H plus plus HHB, Haldane effect. Then NaCl, Na plus plus Cl minus, HCO3 minus diffuses to plasma and Cl minus diffuse to RBC. Chloride shift or Hamburger effect or Hamburger phenomenon. Then HCO3 minus plus Na gives NaHCO3 in plasma. Cl minus plus K giving KCl in RBC. And in the blood of lungs, some other processes are taking place. NaHCO3 gives Na plus HCO3 minus in plasma. KCl giving K plus Cl minus in RBC. HCO3 minus diffuses to RBC and Cl minus diffuses back to plasma. HHB giving H plus plus Hb in RBC. H plus plus HCO3 minus giving H2CO3 in RBC. And H2CO3 Hc2O plus plus CO2 in RBC and CO2 diffuses to plasma from RBC. Now the deoxygenated blood returning to lungs contains NaHCO3 in the plasma and hemoglobinic acid and KCl in the RBCs. When this blood reaches pulmonary capillaries, the whole series of events get reversed. The deoxyhemoglobin reverts to the R form. Hemoglobinic acid dissociates and releases hydrogen ions. KCl dissociates and releases Cl minus and NaHCO3 minus dissociates, releasing bicarbonate ions. So the bicarbonate ions soon diffuses into the red cells, combine with hydrogen ions and form carbonic acid. In return, chloride ions diffuse out to plasma, combine with Na or K and form NaCl or KCl. Carbonic acid soon dissociates to CO2 and H2O. The CO2 diffuses to plasma and gets exchanged for the alveolar O2.